So our big question is, of course, how does this happen? How do we get these horrible, terrifying, antibiotic-resistant bacteria? The mechanism is natural selection. This is the mechanics of how it happens. We're going to talk more about why it happens. And we said the first two, everyone's a little bit different and life is tough. Now we're going to explore the next ones. Individuals tend to have more offspring than are going to survive. So I will read this and then shut up. Individuals tend to produce more offspring than will survive. How many babies, and that's in air quotes, obviously, or scare quotes, does a bacteria want to grow up to have? The bacteria says, I'd like to grow up to have all of them. Bacteria will just continue to divide as long as conditions are right. Bacteria require certain temperatures to reproduce, hence fever, hence refrigerating food. Bacterial populations can grow exponentially. So bacteria don't exactly plan their reproduction. Okay? If conditions are right for them, they will reproduce. And it's largely temperature dependent for most species of bacteria. It can also be dependent on like the pH, the acidity of their environment, but for the most part it's down to temperature. So we haven't talked yet about tolerance curves. We'll talk a lot about them when we talk about ecology. We'll look at them and compare them between species. But basically what it says is there is some range of conditions for every species where life is perfect. Everything is good. Individuals can grow, they can reproduce, the population booms, fantastic. And for bacteria, when they're in that ideal temperature range, individuals thrive, individuals are capable of reproduction. If you cool them down, they can't reproduce. That's the first thing to go. For any organism, when conditions are less than ideal, reproduction is the first thing to stop. So I mentioned my poor little avocado plant here, my poor little stunted avocado plant that if it was just in a tropical forest someplace would be 50 feet tall and bearing beautiful, juicy, fat avocados. It's so stressed, it's never going to reproduce. Conditions here are not optimal in terms of temperature, in terms of soil composition, so it's never going to be thriving enough to reproduce. With bacteria, if we keep them just a little bit colder than we want, even if we don't kill them, we can keep them from growing their populations. So putting food in the refrigerator doesn't kill the bacteria that's in it, and there's bacteria in most of our food, but it keeps them from reproducing. It keeps their populations from expanding, and boy, can they expand. When you have a fever, do you think your body gets hot enough to kill bacteria? You cook your hamburger to what, 160? If you spike a 160 fever, well, after 113 it doesn't matter anymore because you're cooked. So your body is never going to heat up enough to kill bacteria that are attacking you, but it can heat up enough to slow down their reproduction. It can heat up enough that it interferes with their ability to grow their population. Maybe keeps their population numbers stable for a little bit. Gives your immune system a chance to fight. So keeping them outside their comfort range, because when they are in their comfort range, bacterial growth patterns look a little bit like this. That's an exponential curve. And if you've ever graphed y equals x squared, that's what the graph of that looks like. It's called an exponential curve because it is literally the curve given by an exponential equation. Y equals x squared. And that's what bacterial growth looks like under the right conditions for them. So changing their temperature tends to slow down their growth, or it can halt their growth altogether. But it keeps them from doing this exponential growth thing. So a little bit more about how bacteria reproduce. It's called binary fission. Again, I'll read it, then shut up and let you copy, and then we'll talk. Binary fission in a nutshell. Bacteria replicate, that means copy their DNA. Identical, usually, copies of DNA move to opposite ends of the cell. Bacteria divide their cytoplasm. And each new bacterial cell gets half the cytoplasm, one copy of the DNA. Easy peasy. You have 2 new bacteria. So binary fission is a form of asexual reproduction. And you'll note that I wrote that up here. Asexual, meaning that one cell splits into 2 new cells. So if you would take both of your hands, if you're done writing, and you can clap them together in front of you, that is one cell. In an asexual reproduction, one cell splits into 2 new cells. It's a handy little visual. One cell splits into 2 cells. Now, grand and glorious multicellular organisms such as yourself and that avocado tree and an earthworm and a ladybug are all the products of sexual reproduction. So take your 2 fists now out here. In sexual reproduction, 2 cells become 1. So in asexual reproduction, you've got 1 cell that splits into 2 new cells. In sexual reproduction, you've got 2 cells that join and fuse to form 1 new cell that has genetic material from both parents. So how many parents does bacteria have? 1. So we talk about a mother cell and a daughter cell, but there's only 1 parent cell. So shouldn't they be identical? I mean, you're just copying the DNA out of this 1 cell and splitting the cell's material, the jelly stuff, the cytoplasm. What would cause them to be non-identical? Mutations. So I say here identical with a little asterisk because mutations happen. Mutations cause differences between this mother cell and one of the daughter cells. And if those mutations are passed on to the daughter cell, then that daughter cell, and we refer to them only as mothers and daughters, I don't know why, but we do. That daughter cell now has different DNA. And when that daughter cell undergoes binary fission, when it reproduces, it's passing on that changed DNA. Boy, messy writing here, Moser. Cause differences. So mutations in copying the DNA, errors in copying the DNA, can cause differences between the mother cells and the daughter cells. Okay, so bacteria just keep having babies as long as conditions are right. They have lots and 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 lots of babies. Depending on the species, some of them reproduce faster than others. And life is tough. And we see some differences in those babies. Now, we tend to see less genetic variation in species that are the products of asexual reproduction, because we don't have mixing of genetic material from 2 parents. But we do have mutations, so we do have some variation. We've got a hard existence, and we've got lots of babies. That brings us to our 4th point. Guess who survives? Individuals with mutations or features that have some advantage for survival tend to pass on more of their genes. I'll read it and then shut up. Bacteria with a thicker cell wall. Bacteria with a slightly different chemical makeup of their cell wall. Bacteria unharmed by a chemical in their environment, like an antibiotic. If the better bacteria is the only one left, who gets to reproduce? Whose offspring make up the next whose offspring get to make up the population, to make up the next generation? So who's heard natural selection described as survival of the fittest? Okay, it's a common definition. It's something we commonly hear. And a lot of people think like it means, who can do the most pull-ups? Who can run the fastest mile? It's who's the best fit for the environment. And I've got Germex up here too, if you'd like so. It's who's the best fit for the environment. Who's most fit may change depending on the environment. The environment is what drives natural selection. So if only the bacteria that are able to resist an antibiotic are left. Who gets to make up the next generation? Their offspring. Their offspring, which is terrifying. So, if we look at this little bacterial population that I've drawn here, and I don't have a whole lot of bacteria that are resistant. We're symbolizing the resistant bacteria here as the ones that have the thicker cell wall. Though that's not the only way that bacteria can be resistant. We can have bacteria that have a different chemical composition, or that go through a different process to do what's called protein synthesis, to build proteins from their DNA instructions. There are a lot of ways in which resistance can evolve, but I'm going to symbolize the mutants here with a little strip of yellow in their DNA. That's the mutation that gives them some advantage. So when we flood this whole area with an antibiotic, when we flood this whole area with an antibiotic, and it kills off, not all maybe, but a lot of them, and everybody I've just killed disappears. So we only had one of those resistant bacteria in a whole field. Maybe it's 1 out of 100, maybe it's 1 out of 1,000,000. But when we start to eliminate the ones that aren't resistant, and we don't kill them all, but now we have these guys reproducing and forming the next generation. Does the ratio of resistant to non-resistant bacteria start to shift? Yeah, sure does. And when we flood this with antibiotics again, and it kills, again, not all, but a lot of the non-resistant bacteria. And then these guys are the ones that get to reproduce. Is the ratio of resistant to non-resistant bacteria shifting? And in whose favor? We got a problem. We got a problem. How many generations does it take to have a population that's mostly resistant? Well, we're going to watch that MIT video again real quickly, because I just can't get enough of the mega plate. I really can't. I find it amazing. So this is the MIT mega plate video, and then I have a little group challenge for you. All right, so you're going to be doing your group challenge. You're going to each write an individual paper and then collectively write one dossier. And this is not actually showing the tools